Alright, student, good morning. Today we start already the cell cycle to top hat. Today we are going to finish it. I will give you some tips. The top hat exercise 4 is not graded, okay? But you need to do it to understand actually the chapter related to the cell cycle and also to be able to answer the questions in the final exam for the lab. So we already covered the first part about mitosis of the cell cycle. The cell cycle is the stage of the cell cycle from the interphase that represents almost 90% of the cell cycle, the phase G1, where the cells, in all these three, the interphase are three stages, G1, gap1, S synthesis phase, and gap2, G2 phase. That both of them are part of the interphase. Interphase represents 90% of the cell cycle. The rest, the 10%, is what we call M phase or mitosis. What happened during the G1 phase of the interphase? The cell grow, increase in volume, and also productions of a lot of proteins and almost all the organelles or structures that they are inside of the cells are going to be replicated, except one. It's your DNA. DNA will be replicated during the S phase of the interphase, where also the cells continue to grow, right? But the DNA will be replicated in double, okay? During the G2, what's going to happen? During the G2 phase, and that's the end of the interphase, all right? It's a phase that it's growing, right? If it's an animal cell, this is where you remember the animal have a centrosome, and the centrosome has a pair of centriole on it, which are made of microtubules. The plant does not have a centriole, does not have a centrosome, but they do evolve other structure of microtubule to able them to aggregate the chromosomes. Some other structures play this job. We don't really know which one, but they do have microtubules in the plant cells, but they don't have a centrosome, they don't have a pair of centriole. So if it's an animal cell, during the G2 phase, the centriole that has been replicated during the G1 phase are going to start being separated by the end of G2, right? And also what's going to happen during the G2, it's going to make sure that everything, any organelle inside of the cells is seen double, is looking double. Double of the DNA is replicated. Everything has to be replicated during this phase, making sure if it's not being replicated, you will send it. For example, if the DNA is not normally replicated, you will send it back to the S phase. If, for example, I will say let's see, ribosome is not well replicated, you will send it, you got it, to the G1 phase. So G2 is a very, very important phase. And as I said, the interphase is very long, 90% of the cell cycle. It's between 20 to 24 hours. This is how long this phase is in any cell cycle time. Talking about time, it's about 20 to 24 hours. It's a long time. During this phase of interphase, they are preparing the cell to enter the M phase, which is the cell division, which is the mitosis or meiosis, as we are going to see later on. So they enter to the mitosis phase. Mitosis phase have four metaphase phases. They have a prophase, metaphase, anaphase, and telophase. And after that, the cell either can exit the cell cycle, all right, to do whatever job she have to do, or some of them can also go into, again, replicate around the cycle. Sometimes the cell will not enter, will come to enter the G1 phase, but once at the G1 phase, it's going to rest. We say that the cell enter a phase that we call, this phase could be either latency cell that is slowing down, it's a G0. And then after that, some signal will be received and she will continue her process through the cell cycle. Or this cell can stay during the G1 phase until it died. And when it died always by programmed cell death, which is what we call apoptosis. All right. So let me keep going now. We are going to look at all the phases of the mitosis M phases. All right. The end of the M phase, which is the mitosis phase, it's what we call cytokinosis. Cytokinosis is it's separation of the cytoplasm. Cytokinesis means separation. It's when the cytoplasm are going to be separated and therefore separate the cell into two identical cells. So this is cytokinosis, and it's always a start before even depend on the cell. Sometimes it starts before even the telophase finish, and sometimes you wait until the telophase finish before the cytoplasm starts dividing. It depends on the cell, okay? But it's always by the end of the telophase. So what's happened during the first phase of the cell cycle, the mitosis phase? You remember that my chromosomes. Remember that chromosome is a DNA filamentous DNA and add some proteins on it, all right? When the chromosomes, when those are divided, we call them chromosomes. And when they are just sitting together, like during the interphase, they are called chromatin. Chromatin, okay? So during the phase, what's going to happen? You remember that the chromosome has been replicated during the S phase of the interphase, all right? So we have what we call chromatid sister. Chromatid sister are two chromosomes that they are attached by their centromere. I explained to you what is centromere during my lecture. Centromere is this neuro that is separate each chromosome on short arm and the long arm. So each chromosome has this centromere. And when he got replicated, we don't call it chromosome anymore. We call it chromatid, and they are sisters because they are like each other. They are real twin sisters. Let's look at it, say it this way. So they still attached by their centromere. So the sister chromatids, this is when the chromosomes start to condense, making them visible. We are going to start seeing them. And remember that I told you that during the G2 phase, the late G2 phase, the centriole, pair of centriole are going to start separated, and they are made of microtubules. And they are like a spider with a web. Once he is moving, he's making this web, moving this web. And they are what we call aster, like stars over here, asters. So they are going to begin forming this, what we call mitotic spindle. Mitotic spindle, that they are what? They are just microtubules that they grow out from this mitotic spindle. They are microtubules that they are coming out from this pair of centrioles that exist in the centrosome. This is for sure what I have over here. It's an animal cell, because not only the form, the geometric form is around, but also the presence of this pair of centrioles. Okay? By the late phase of the prophase, to be honest with you, it's very hard to differentiate between the late phase of prophase and the beginning of metaphase, because the nuclear envelope got completely, the nucleolus disappear, first of all. The nucleolus is gone, right? And the nuclear envelope start to be fragmented. It is willing to disappear. It's going to be fragmented. Okay, so during the prophase, not only I have chromosomes that are going to be condensed, I don't call them chromatin. Chromatin is during the interphase. Remember that. Chromatin is the chromosome that is not divided. Yeah. Once it got divided, we call them chromosome. The chromosome condense. They are chromatid sister because they got being replicated. But I cannot see the chromatid sisters. I cannot see them. You know where I am going to see them beautifully, very nicely chromatid sister attached is during the metaphase plate. It's during the metaphase. This is chromatid sister. This is one chromatid sister, and this is another chromatid sister. And I told you this is the centromere. This is the centromere. So what I want you to do, I am going to ask you to go to your lab manual, and in the page, as I said, let me look at the page that what I have is where the cell cycle, I call it lab mitosis, and it's in the page 39. I want you to look at those questions in the page 42. Look at the manual. You have already picture and illustration of a prophase. I want you to get familiar, to draw it, do it. I know I am not grading this, but it will help you not only during the exam lecture, but also the lab exam. Do that, please. Draw a prophase phase. Okay? Then we move in. The nucleus envelope is completely fragmented. You see, it's just fragment, and we can see the chromatid system very visible. It seems like this is a prometaphase late prophase, this phase, right? Why it's not a metaphase? Why it's not a metaphase? It's not a metaphase because those chromosomes have to come a little down on the middle or the diameter of whatever it's your circle over here, in the equator of your cell. That's equator of the cell, we call it metaphase plate. And this is exactly what I am seeing over here. They are laying down. And look at the mitotic spindle. Look at those mitotic spindle. You remember the centriole that they start moving separated at the G2 phase, and they start forming this mitotic spindle. They're in the prophase, and it's completely formed. They're in the prometaphase. It's completely done in the metaphase. Completely. It's completely done. They're in the late prophase and beginning of prometaphase. The nuclear envelope doesn't exist anymore, and you will see the content of the nucleus that is called nucleus before. This is where that contain my chromosomes is completely mixed in the cytoplasm. It's gone. My nuclear envelope disappear. All right, look at that. It laid down in the middle. This is an animals, again, not only the geometric form, but also the presence of the paracentriole. This is a plant cells. This is very typical plant cells, and this dark green over here, it's your cell wall, all right? And you see that the nuclear envelope is completely gone, guys. Did you notice that? Completely gone. It's not there anymore. Let's look at this mitotic spindle. And the chromosomes, hold on. This is one chromatid sister, this is another chromatid sister, and they're attached by their centromere. Exactly, centromere. And each chromatid sister, right, their centromere, it's in one pole. This is one chromatid sister in one pole, and the other chromatid sister is facing the opposite pole, where the pair of centrioles are located. All right, well, let's look at this mitotic spindle. It's amazing, it's beautiful. Look at those mitotic spindle. I have two types of mitotic spindle over here. Remember, those mitotic spindles are formed just by microtubules. These microtubules came out from the centrosome, which are pair of centriole. Remember, centriole are made of 9 it's a triplet of microtubules. Remember that when we study the cell structures, all right? So look at this mitotic spindle. We have two types, as I said. I have one mitotic spindle attached. It's attached to the centromere, isn't it? Right? Look at this, right? This dark blue over here, right? The sky blue is not attached to the centromere. And actually, they are not attached to the centromere. They are attached to a molecule that has the same location than the centromere. And this molecule we call it kinetochore. Kinetochore. I'm seeing if I can find the word somewhere. No, I don't have it, but I will try to write it down. Hold on, let me write it down. Let's see. I am at this slide. T-I-N-E. I'm spelling it right. This is an N, guys. Kinetochore, right? And therefore, we have microtubules kinetochore, the one that they are attached to my kinetochorolicles. Why my little hand disappeared? There you go. Those dark blue over here, that's called kinetochore microtubules. The job for those kinetochore microtubules is to pull, to separate the chromatid sister from each other. It's to pull them to the opposite poles of the cell. It's to pull them out. It's to separate them out. Non-kinetochore microtubules, their job, therefore, is related to the cytoplasm. They are going to elongate in the same times. Those are going to pull those with the presence of proteins, of course. They are going to pull those chromatid sister and separate them from each other. Those are going to elongate the cytoplasm so it can have better space. I don't like this picture for the simple reason they are showing like there are no mitotic spindles. There are mitotic spindles over here too in the plant cells, but other structures play a role on forming those mitotic spindles of the plant. I really, that was very, very naive pictures over here. I don't know why they didn't put mitotic spindle. They have some spindles, but I said we don't really know which structures they are because we have cytoskeletons made of microtubules on the plant too. So we do have some mitotic spindles over here too. Anyway, You know how long is going to be the prophase versus the metaphase phase in time during these divisions? As I said, the interface, it's almost, as I said, 20 to 24 hours. Metaphase phase, it's about minutes. The prophase maximum can go. This is one of the longest phase of the M phase, 1 hour. It go between 30 minutes to 60 minutes, right? The metaphase, you know how long? 2 to 10 minutes. 2 to 10 minutes. Look at that phase, 20 to 24 hours. I came up to the prophase maximum 1 hour, 30 to 60 minutes. I came up to the metaphase 2 to 10 minutes. Then the anaphase is one of the quicker phases. I don't really know why, but my suggestion is because of the job of kinetic, of the movement of the protein motors over here. It's only maximum they can go, the anaphase is 3 minutes. It's very quick phase, very, very quick. And then after they got separated, which phase? The last phase, which is, it doesn't, the last phase. Why this slide does not appear? That's the anaphase. As I said, look at that, the kenotic or microtubules, they are pulling the chromatid sister. Look at that. That's non-kenotic part. Their job is to elongate. It's like an elastic, elongate the cytoplasm. But again, I don't like these pictures because they are mitotic spindles over here. The only reason they didn't put it so you can understand, because we really don't know the spindles originate from where. I'm sure they have some evolved structure that make those mitotic spindles. Again, after that, what we have, telophase. Telophase is like the opposite of the late prophase. It's the opposite of it. The nuclear envelope starts appearing, right? And the chromatid sister now become chromosomes. That's it, right? And the mitotic spindle are reabsorbed. They start to be disappearing. All right. And now what I have, look at that. I have a separation of the cytoplasm. Is going to be cut. This is by cleavage ferro. This is what we call cytokinosis, right? Cytokinosis. And the cells, if you look at them, they are identical to each other, and they have 1, 2, 3, 4, 5. I have 6 chromosomes. I end up having 6 chromosomes, and my cells in the beginning start with 6 chromosomes, the mother cell, right? For the plant cells, this is the same things that happen. You look at that, they are the same phases, okay? All go through the interphase. Then the M phase are the same also. The only difference is the way the cytoplasm is going to be separated. Here is by cleavage ferro. Here is by what we call a cell plate. Golgi apparatus will secrete some vesicles over here, to form a new cell wall. And once the cell wall is formed, it will separate the two cells into identical cells that have also 6 chromosomes, as well as the mother cell. And in the end, I have my 2 daughter cells, and the nucleus start to be appeared. You see the red one, that's the nucleolus. And they have 2 cells with the nucleus, etc., etc. All right, guys. So really, really what I want you to do, I want want you to look at the page that is, hold on, page 42 and 43 in your lab manual, and you have 44 in the lab manual, and you have 45, 46. So from 42 to 46 in your lab manual, I want you to answer those questions. If you feel confident that you answer all the questions correctly and you don't need my help, it's fine. But if you feel like, hey, I do need Dr. Carver assistance, you can always email me and they can meet with you and I go over those questions with you. So you have also to answer those questions. Why mitosis is essential? Why? I know the answer? No, I don't. Let me look at this lab from the beginning. Let me go and look at the lecture. I love when a lecture is parallel with the lab. I still didn't get it. Well, write it down. Contact Dr. Carver. Were you able to see the same phase mitosis in the plant and the animal? Am I seeing all the phases? Is the plant self-divided in the same way than the animals? Kind of, yes. Except we have to make a difference. Except that the way the cytoplasm divide, the cytokinosis, animal is by cleavage furrow, plant is by cell plate. So we do have for the plant and the animal cells, we have a prophase, metaphase, anaphase, telophase, and chromosome spindle fiber. The plant cells does have cell plates will form the cell wall that separate the two cells during the telophase. For the animal cells, we have centrosome and centriole that form asters, and therefore this aster will be developed into microtubule fibers or spindle, mitotic spindles, and they divide, their cytoplasm divide by cleavage furrow. And the cleavage furrow separates actually the cell membrane that will separate. The other one is the cell wall. This one is the cell membrane that will separate the two cells during the end of the telophase. They are asking you also during mitosis what structures are absent in animal and are not found in plant. Listen what I just said. You will have the answer. In the telophase, the daughter cells are pinched off in an animal cells. This is what we call pinching. It's like you're taking all of you're making a bread and you put like pinching, whereas the plant cells are separated by the cell wall. And which structures are present in the animal, you know it very well, is the centrosome with the pair of centriole, right? Which structures are the same during mitosis? They are both, they have everything. They have chromosomes and mitotic spindle. This is why I didn't like these pictures that I told you, naive of the plant cells that they are not showing those spindles. It's very naive. It makes me like, oh God, no. All right. This is why it's very important that I go over each the text with you. How does the separation cell differ in the plant cells? We keep the same thing. They said, how do the pole of spindle differ from the animal cells and the plant cells? Where in the animal, it came from the centriosome, which have a pair of centrioles. So it came actually from the centriole. And we know that the centriole is 9 plus 3 mitotic microtubules. For the plant, we really doesn't know. We know that they have some other structures that do that. And trust me, you are completely done. I mean, and they want you to just write in piece of paper what happened during the interphase, what happened in the early prophase, what happened in the light prophase, which is also called the pro-metaphase, or what happened during the metaphase, what happened during the anaphase, what happened during the telophase and cytokinosis. And in the plant, I want you to do the same things. Write it down. Trust me, you must write down. Don't be lazy. Not because I am not grading this exercise that you are going to be lazy. If you want to understand my lecture, do those things. They will help you with the questions. All right? They will help you with the exam, lecture exam, and the lab exam. And please do not text me after the exam. I didn't understand. I am here. You can text me. And don't start your exam 2 hours before the due date. In case you have some technical issue and you have to contact the proctorial people. I realize one thing. I realize that proctorial, some computers, especially when you have antiviral things, you were rejected. So all the time before any exam, you have to make sure. I'm saying it. And because I'm saying it, I'm not going to reopen exam because of this technical issue anymore, because I'm saying it. I am asking you to verify and update your Chrome and your proctorial method every time. You have also an exam that I post. It's a test. Test yourself. Test yourself. Is the proctorial method? It's just one question. Is your proctorial method working? It's not a graded exam. It's just making sure that your proctorial method is working. So all the time you have to start it. Give your time. Give yourself time. Even myself, I have all the time to update. It's not update. It's actually to fresh up my Chrome with my proctorial method because of the type of antiviral that we have in my computer is very, very protected by my university. So yes, and sometimes it's happened. When he asks you password, just submit. Keep submitting, and you will see the exam appearing. You don't need a password. I have no control on the password. Password came from the proctorial people. All right, and that's it. You have here. I will leave that here in the your blackboard over here. So this is just to say, use the information that is there in this lab. You have to answer those questions. What happened to the nucleus during the interphase, prophase, metaphase, anaphase, telophase? Prophase, metaphase, anaphase, we don't have nucleus anymore. It's completely, his content completely inside of the cytoplasm. It starts to be formed when the nuclear envelope formed. The nuclear envelope, it's very important to make the nucleus have a compartment that we call nucleus. And what happened to the chromosome? Well, after when they enter the division, it's during the interphase, it's called the chromatid. It's called chromatine. Mitotic spindle. Amount of the time, I told you the interphase is one of the longest of the cell cycle, 20 to 24 hours. Prophase is one of the longest of the mitosis. The shorter is the anaphase. The shorter is the anaphase. Telophase, I would say between 3 to 12 minutes. Anaphase is between 2 to 3 minutes, which is very short. Metaphase, it's between 2 to 10 minutes. All right, dear, let's end up our cell cycle lab for today and the tips how we study it. If you have any questions, you are more than welcome to be in communication with me. Please listen and watch those videos. Thank you so much. Stay safe.